Dear students, I will represent to you lecture muscle tissue. In general, muscle tissue classified into three types: skeletal, cardiac, and smooth muscle. Also, we was classified by contraction, voluntary, and involuntary. Voluntary skeletal muscles or involuntary cardiac and smooth muscles. On its scheme, very nice visible differences between types of muscle tissue. Striated, non-striated muscle. What does it mean, striated? We have striations in muscles, very nice visible on slate, histological specimen. And skeletal muscle compound includes some striations, black and light lines, and similarly positioned lines in cardiac muscle. Together, skeletal and cardiac muscles are striated. Smooth muscle, non-striated, because position of myofibrils not parallel and not version on slate, black and light lines. Skeletal muscle includes structure unit, its fibers, muscular fibers. In general, has cylindrical shape, very long, maybe sometime near 30 centimeter, and diameter may be situated by position of different types of muscles from 10 to 60 micrometer. I told before about composition of skeletal muscle, which include more fibers, muscular fibers. Which is structure of fiber? In general, fiber include syncytium, it's myosyncytium, which form after fusion of more cells, and in result, inside in syncytium present more nucleuses, and each nucleus includes by after fusion of each cell. In general, maybe 200 nucleuses in myosyncytium. Myosyncytium surround and covered by sarcolemma and include inside cytoplasm and myofibrils. Sarcolemma similar to plasma membrane of cells, but include not just plasma membrane. Plasma membrane plus myosatellitocytes, it's a special cells which include just one nucleus and then cover of its structure by basal membrane. In general, it's sarcolemma. Sarcoplasm has names is cytoplasm of fiber, and in sarcoplasm present more glycogen inclusions, also present myofibrils, which include inside myosin and actin filaments. Myofibrils include inside more sarcomeres. What does it mean, sarcomere? Sarcomere is unit of contraction. It's a distance between two Z lines or two Z discs, and its distance include myofibrils, its actin and myosin elements. Myosin surrounded by 6 actin myofilaments. Its composition very strong, and parallel layers of myofibrils formed sarcomeres. Sarcomeres in result form light and dark lines in striated skeletal muscle. If you will see a histological specimen skeletal muscle after staining of iron hematoxylin, you can see some elements find of dark lines and light lines parallel and it's form some structures. Dark line or dark bands are called A bands or A discs, and light bands are called I bands or I discs. Students, it's a good slide. Show to you photo from electron micrograph. Very nice visible on photo Z lines, location of Z line. It's a dark staining line, and between two Z lines stay sarcomere. Sarcomere include more dark staining A zone or A band and light staining I band. And in central part of sarcomere present special M line. Very important composition of actin myofilaments. Each actin myofilament consists of double helix of F-actins molecules composed of G-actin monomers, tropomyosin and troponin. Myosin molecules consisting of two globular heads and a rod-like portion constitute myosin myofilaments. And in general, in scheme, it's a vision by head and special tail. It's a good slide show composition of actin myofilament. You see monomers, actin monomers, it's a molecule, it's a protein molecule, and together with troponin and tropomyosin formed special actin filament or thin filament. It's always in slate light staining filament. And position of elements, you can see vision, numbers, and how many monomers, actin monomers include in filament, and location of troponin, blue color, and tropomyosin, yellow color on its picture. It's a good scheme from your textbook. You can see a structure of skeletal muscle. To start from muscle in general, its organ, and next include muscular fasciculates, which include muscle fiber inside. In every muscle fiber includes sarcomeres. It's a contraction unit of contraction. Each sarcomere includes lines or zones, dark staining and light staining. And that line is a central part of light staining I band, and H zone is a central part of A band, dark staining. And here includes some elements of actin myofilament with a vision on its slate and myosin filament. Please, you can see its picture in textbook and recognize the elements of myofibrils. Very important mechanism of contraction. It's a special model, sliding filament model, which includes a joining between elements myosin and actin filaments. First of all, the first step of contraction will be increased calcium inside in sarcoplasm, and after it will be signal to change location of troponin and open tropomyosin in actin filaments. Then after, head of myosin joined with tropomyosin and to start movement to change location of its head. In result, actin filaments will be moved to central part of sarcomere, and after a strong contraction, we cannot see on slate light bands, just dark bands, because actin filaments includes in central part of sarcomere and close myosin filaments area of eye band. It's a good scheme, show to you a process of contraction, and you can see on its picture very important calcium and ATP, because process of movement of head of myosin and the next unjoined head of myosin from actin filaments is energy dependent process. Very important height level of calcium, I repeat to you, and ATP. In its case, in muscle fibers present more mitochondria. Very important sarcoplasmic reticulum. Sarcoplasmic reticulum provides very important function in muscle fiber and in general muscles. Also, which save of ions of calcium. And ions of calcium very important to start process of contraction. Sarcoplasmic reticulum includes more calcium ions. It's a special process of deposition of calcium, and it's a special smooth endoplasmic reticulum. In general, in its scheme, very nice visible composition of sarcoplasmic reticulum. We have inside in sarcoplasmic reticulum special triad, which include T-tubule and cisterns, terminal cisterns. In terminal cisterns save calcium. Terminal cisterns in central part of sarcomere form special joining and in result hollow some present, and in central part of A band present more large its hole. It's very nice visible. Yellow color in its picture is T-tubes and cisterns, and in general sarcoplasmic reticulum is blue. Look very attentive. VA is located of triad. Triads lie between two bands, A and I bands. In general, in one sarcomere of skeletal muscle present two triads. Activation of sarcoplasmic reticulum to start from depolarization of plasma membrane. It's a process influence on T-tubules, and then T-tubules will be regulate releasing process of calcium from sarcoplasmic reticulum. Sarcoplasmic reticulum release calcium and to start process of contraction. And before I told about special mechanism of contraction, and the first step is release calcium. It's a good scheme. Stay in your textbook. You can see its scheme. Please look very attentive of process of contraction. Here present scheme location of nerve ending release impulse to special motor end plate, and its influences of electrical impulses goes to muscle fibers, and then in muscle fibers to start process of contraction. And the first step is release calcium in sarcoplasm. After that, to start process contraction, and you can see on its picture very nice visible contracted sarcomeres. In contracted sarcomeres, not visible I band, just A bands, dark staining bands. On its slide, you can see test control question, and please look and recognize correct answer. It's to individual work. Cardiac muscle. Cardiac muscle forms special myocardium. It's a middle part of wall of heart. And in central of myocardium lies special cell. Its cell has name cardiomyocyte. Cardiomuscle not form fibers of muscle and just cell cardiomyocytes form myocardium. Formation of cardiac muscle to start from mesoderm, it's a ventral part of mesoderm, before formed myoepicardial plate. From myoepicardial plate will be formed myocardium and epicardium. It's just ventral mesoderm visceral layer. About size. You can see on its slate size of cells and near 15 micrometer in diameter each cell and more long 80 micrometer, and in central part present single large oval nucleus, and in sometime in cell may be present two nucleuses. It's good scheme structure of myocardium. You can see yellow color its nucleus and size of cell. Black line you can see not strong line, some form zigzag, and its line it's a junction between two cardiomyocytes. Its junction has name intercalated disc. Intercalated disc is a special junction between cardiomyocytes which includes some different types of cell junctions. It's a desmosome, it's a gap junction or nexus. Gap junction formed after communication between cells. It's a communication formed by special protein connectin and which form connexons. And the connexons together form strong communicative between cells. Very important gap junction which because provide calcium from one cell to another. In cardiomyocytes, not more large as sarcoplasmic reticulum and calcium migrate from one cell to another, which provide contraction in 1 minute more cells, and in result contract more cells in one moment. In general, in 1 minute in adult, near 70 beats. Students, it's a very good scheme. Show to you structure of intercalated discs, and here you can see position of intercalated disc and structure of its disc. Present some communicative elements, and very important desmosome, gap junction, and tight junction. On its slate, you can see a photo from electron microscope and position of intercalated discs. And it's are lines, dark staining, with joint two cells. Third type of muscles has name smooth muscle. Smooth muscle are called non-striated, involuntary muscle, which provide contraction more slowly and not more strong, appositive of striated muscle. And skeletal muscle will be regulated by motor nerve endings, and which regulating by brain and is voluntary. Smooth muscle involuntary and more regulate from vegetative nerve system, sympathetic and parasympathetic. Main element of smooth muscle has name smooth muscle cell. Cell form a junction between one by one and in result form fibers. But it's not correct name fiber because fiber include more nucleuses in skeletal muscle. Here it's a will be junction between cells, smooth muscle cells, communicate and form long structures. Variable cells from 15 to 500 micrometer. Smooth muscle joint and form bundles or fascicules and with special direction. Direction may be circulatory or longitudinal, and also a smooth muscle present in tubular organs. Tubular organs, it's a special type of organ which include cavity inside like tube and wall of tubular organ forms by some layers. It's mucous layer and the muscular layer and serous or adventitial layer. Muscles formed tubular organs and between muscle cells present connective tissue with blood vessels. It's a very good photo from light microscope. And on its photo, direction of smooth muscle cell difference. Here you can see if it's a longitudinal line on transverse section will be visible just area of cell, it's a diameter of cell in central part or peripheral part. If it's central part, visible nucleus inside. Oval structures in nucleus in central part. Okay, next direction it will be circulatory on its picture. Visible all line of cell. Cell has spindle-like shape, and in central part present oval nucleus. Students, it's a good scheme, location of smooth muscle cells and different vision of smooth muscle cells to concern special sections. Here, if it's transverse section, very nice visible parts of cells. If it's section line on central part of cell, visible nucleus in central part. Oval shaped structures in central part nucleus. If its section will be on ending of type of cells, ending part, visible small areas, small round structures without nucleus. On longitudinal section, very nice visible cell. It's a spindle-like shape of cell. In central part, present nucleus, oval shape. Mechanism of contraction in smooth muscle is different from skeletal muscle and cardiac muscle. Its difference joined with location of myofibril. Myofibril not form parallel layers, not form sarcomeres. Here will be location of actin and myosin filaments network form inside, and actin filaments may be joined with plasma membrane. In its case, present special protein alpha-actinin. What's happened with smooth muscle cell in period of contraction? The first, after influence of nerve ending signal, to start process of depolarization of plasma membrane and release calcium from special carbules. Carbules, it's an invagination of plasma membrane inside in cell which provide save of calcium. After increased concentration of calcium inside in cytoplasm, will be start process of phosphorylation of myosin elements, and after phosphorylation to start process of contraction, and very great difference between skeletal muscle and smooth muscle, it's a not present troponin. After phosphorylation, a head of myosin will be joined with actin filament and after use of ATP energy to start process of movement and in result contraction of cell. Tubules save of calcium, its invagination of plasma membrane in central parts of cell in cytoplasm and after releasing of calcium from tubules inside in cytoplasm to start process of contraction. Contraction in smooth muscle cell very interesting because it's not really similar to contraction in skeletal muscle because in skeletal muscle contraction always decrease size by long of muscle fibers. Here it's another situation. After contractions, cells stay maybe not short, but sometime more long, but very interesting changes in nucleus inside in cell. Nucleus stay not oval shape, maybe sometime form spiral and spiralization of nucleus. Here you can see scheme. What happened with smooth muscle cell before contraction and after contraction. Very nice visible black lines, its location of actin filament and points, black points, it's a special alpha actinin which joined with plasma membrane and joined with actin filaments and in result to provide very interesting contraction of smooth muscle cell. Students, it's a last slide. Thank you to your attention. Goodbye, good luck.